A very warm welcome to all of you guys. Welcome back in another video tutorial of our enzymology lecture series. So today we will going to talk about a special topic on enzymology, that is zymogen. So zymogen, which is the name for precursor of the enzyme, or you may say this as pre-enzyme. Okay, so what is this? So this precursor of the enzyme or zymogen is the inactive form of enzyme. There are many enzymes present in our biological system that initially synthesized in inactive form, and then by the help of some type of cleavage it will be converted into its active format. Okay, the idea is when that enzyme will be synthesized, its active site will be blocked. Okay, and after that, with the help of cleavage mediated by some type of enzyme, its active site will be exposed due to some sort of conformational changes. Okay, that will convert it into active form of the enzyme. And these inactive enzymes are known as zymogen. Okay, there are various type of proteolytic enzyme present that are zymogen in its initial form when they are synthesized. Okay, for example, trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase are the examples of zymogen, and they are converted into its active form after being cleaved with the help of various type of enzymes. Okay, for example, in duodenum, trypsinogen, chymotrypsinogen, proelastase and procarboxypeptidase zymogens are converted into its their active format. Okay, which is trypsin in case of trypsinogen, elastase in case of proelastase, chymotrypsin in case of chymotrypsinogen, and carboxypeptidase in case of procarboxypeptidase. Okay, if you have noticed, trypsinogen is activated by enteropeptidase, and now this trypsin is activating other enzymes, right, in the duodenum. Okay, so now here we will going to see how a enzyme from its zymogen format converted into its active format. So here we will going to consider the example of chymotrypsinogen. Now before going to talk about this, there is a one question that is going on in my mind: why this type of enzymes are synthesized in the form of zymogen? So the answer of that question is probably due to the source or the origin of those type of enzyme. I mean to say that the organ where this type of enzymes are synthesizing should contain other type of things that may be sensitive to this type of enzyme. So for protecting those type of things, this type of enzymes are synthesized in its inactive format, right? So that that particular organ should be intact. Okay, so that may be a possible reason why zymogens are synthesized, or why some type of enzymes are synthesized in their inactive form. So here we are now back to the topic. Here we are talking about chymotrypsinogen. So chymotrypsinogen is a protein of 245 amino acids, which is a single polypeptide when it was in its zymogen form. And as we have seen in the previous diagram, we have seen that chymotrypsinogen will be activated by trypsin, right? So trypsin will cleave the chymotrypsinogen, converting it into two polypeptide containing pi chymotrypsinogen, which is a little bit active, and then from that again trypsin will work and giving us alpha chymotrypsin. Okay, and here now it will be consisting of three polypeptides that is connected by two disulfide bond. Remember, from the very first inactive form or zymogen form of chymotrypsin, that is chymotrypsinogens, there are five disulfide bond present, right? First it is converted into pi chymotrypsin with the help of the trypsin enzyme, and then from that it will be converted into alpha chymotrypsin, which is a 3 polypeptide linked by two disulfide bonds, right? Which was already previously present in the chymotrypsinogen. So that's the idea. So that is our concept of zymogen. Okay, so thank you for listening to this class.